Look at this structure. This is a complex microscopic network of glowing nodes and physical threads, seamlessly self-assembling into a highly organized geometric pattern. It demonstrates how biological systems operate as deeply interconnected networks rather than simple chemical combustion engines. For decades, nutrition science has treated the human body like a simple furnace. In this model, food is reduced strictly to a chemical substrate or a unit of caloric energy to be burned. A strictly caloric view fails to explain how the body maintains its structural and electromagnetic integrity under stress. A more accurate model is the Food Coherence System, or FCS. In biophysics, coherence refers to the physical stability, structural organization, and functional alignment of interacting biological systems. It measures how well local cellular events stay synchronized with the rest of the body. This requires us to look at food as an informational input. Its structural matrix either integrates smoothly into the body's internal architecture, or it introduces physiological noise that forces the system to compensate, eventually driving systemic failure. To survive and function, a living organism requires a precise internal coordinate system. This spatial map is established from the earliest stages of embryogenesis and dictates where every cell belongs. This early positional order is created by morphogen gradients. A morphogen gradient is a concentration of signaling molecules that diffuses across an embryo, giving cells specific spatial coordinates for differentiation. This animation shows a 3D isometric wireframe of a human silhouette, representing the distributed fascial matrix. The initial spatial order of the embryo scales up into this adult form through a body-wide continuum of connective tissue. This network operates on the principle of biodynamic tensegrity. Biodynamic tensegrity is a structural model where continuous tension and discontinuous compression allow a system to distribute mechanical load and transmit physical force across its entire geometry. Mechanical form acts as the physical infrastructure of the body. It actively aligns local cellular events with the whole system regulatory architecture, ensuring that physical stress is managed evenly across the network. A solid wireframe cannot function in a vacuum. It is surrounded and interpenetrated by a fluid environment. Inside the body, water does not behave like the inert bulk solvent you would find in a glass. As it approaches biological interfaces, its physical properties change. This diagram adds a fluid dynamic layer to the 3D fascial wireframe we just saw. Notice the distinct, highly organized geometric alignment of molecules immediately adjacent to the structural surfaces. This is interfacial water, or easy water. It adopts highly ordered states and altered charge distributions when it sits near physical boundaries. In this ordered state, the fluid acts as a biological semiconductor. This allows for rapid proton conduction, moving charge much faster than slow, random diffusion would permit. A highly ordered fluid medium preserves informational stability. It eliminates transport delay in metabolic signaling, allowing cells to communicate rapidly across the entire physical matrix. Moving information through this fluid matrix requires an electrical hierarchy. The body uses multiple layers of electrical signaling for continuous biological regulation. Pulsing electrical pathways traveling along the structural human matrix represent active bioelectricity, which relies on ATP-dependent ion gradients for cell-to-cell signaling. A second, passive regulatory layer, piezoelectricity, generates electrical activity through mechanical deformation of tissues without requiring ATP expenditure. Piezoelectricity serves as a direct bridge. It translates pure mechanical force, like the compression of a bone or the stretching of a tendon, into adaptive cellular signaling. Physical movement and structural load are informational events. Without physical movement, the organism loses the critical electrical communication required to trigger structural remodeling and tissue repair. Beyond electricity, the system utilizes an optical signaling layer. Living tissues continuously emit and respond to ultra-weak light, known as biophotons. These optical dynamics serve as a candidate indicator of tissue order. Healthy, organized tissues display regulated, stable emission patterns, while stressed or damaged tissues leak erratic, irregular signal noise. All of these signals, the mechanical tension, the fluid charge, the electrical gradients, and the optical emissions, converge on the cellular nucleus. Together, they dictate gene expression. The nucleus uses mechanisms like DNA methylation for selective control. DNA methylation is an epigenetic process where chemical tags are added to DNA, keeping necessary genomic regions active while silencing others based on the upstream structural signals the cell receives. Gene expression is an adaptive, real-time response to the structural and electromagnetic coherence of the cell's environment. The genetic code is only read selectively, guided by the physical state of the network. This finely tuned biological coordinate system faces a severe stress test when introduced to ultra-processed foods and synthetic additives. This diagram highlights the intestinal barrier. On the left, an ordered gut matrix processes structured natural food. On the right, synthetic additives dismantle the barrier. These chaotic vectors cascade upward, degrading organized fluid and electrical layers into fragmented noise, contaminating the system. A constant barrage of low-level additive burden creates an internal environment known as an industrial swamp. The fluid matrix loses its order, increasing physiological noise and delaying cellular transport across the body. Industrial food may possess perfect synthetic order on a supermarket shelf, uniform texture, stable color, and long shelf life. But inside the organism, it functions as a potent communication disruptor that severs systemic alignment. When the gut interface fails, the burden shifts downstream. The liver, which serves as the central processing node for the body, becomes severely bottlenecked by inflammatory and xenobiotic noise. When total filtration capacity is exceeded, the body employs distributed survival logic. Distributed survival logic is an adaptive triage strategy where the organism prioritizes short-term vital organ viability over long-term systemic coherence. This diagram illustrates the resulting physiological adaptation. The central liver node flashes an overload state, routing dense, toxic input nodes away from vital organs and locking them into expanding, isolated adipose compartments on the periphery. This creates the fat sarcophagus. Under this burden, adipose tissue ceases to be a dynamic energy reservoir. Instead, it becomes a protective containment zone for lipophilic toxicants that the liver cannot currently process. The accumulation of intractable fat is a biophysical sequestration strategy. The body deliberately isolates these inputs to protect the core network from lethal signal noise. This explains why rapid weight loss interventions often fail or trigger severe physical rebound when attempted within a burdened state. Aggressively mobilizing sequestered fat into a system that still lacks fluid clearance and structural integrity simply refloods the network with the exact noise it was trying to hide. Look at this macroscopic view of an intricate, highly organized geometric biological structure assembling itself smoothly. This structural precision highlights the divergence between simple caloric accounting and the multidimensional reality of the food coherence system. Metabolic health resides in the organism's capacity to preserve uncorrupted communication across every mechanical, fluid, and electromagnetic layer.